This is the last section in Chapter 11. A little bit longer screencast, a little bit more complicated. We'll talk about it tomorrow in class, anything you need to clear up. This is about meiosis and gene linkage and gene mapping. All very interesting things. Remember that chromosomes are the carriers of genes. There are hundreds to thousands of genes on every single chromosome, and those genes are located in specific positions along the chromosomes. If we're talking about a fruit fly, there are 8 chromosomes as shown in this figure. You see that in the nucleus. 4 of the chromosomes come from the male parent and 4 come from the female parent, sexually reproducing organism. These 2 sets of chromosomes are called homologous chromosomes. Each of the 4 chromosomes from a male parent has a corresponding identical chromosome from the female parent. So you see these grouped together. The green chromosomes here are homologous. These little purple guys here are homologous chromosomes. The red ones are homologous. And then the orange ones are also called homologous chromosomes. A cell that contains 2 sets of homologous chromosomes is called diploid. And we represent diploid by the symbol 2n. So the diploid number for a fruit fly is 8. 2n equals 8. N represents twice the number of chromosomes in a sperm or egg cell. That should say 2n represents twice the number of chromosomes in a sperm or egg cell. N is the number of chromosomes in a sperm or egg cell. Diploid number for humans is 46. You'll get to know that number pretty well. So we have 23 pairs of chromosomes in humans. Some cells, like gametes, contain a single set of chromosomes. Those are called haploid. The gametes of sexually reproducing organisms are haploid. In fruit flies, the haploid number, represented by the letter N, is 4. Diploid number is 8. Divide that by 2. The haploid number is 4. Meiosis is the process by which the number of chromosomes changes from diploid to haploid. The way this is done is the separation of homologous chromosomes in a diploid cell. There are 2 divisions in meiosis called meiosis 1 and meiosis 2. By the end of meiosis 2, that product is 4 haploid cells. So prior to meiosis, interphase 1 is pretty much the same. Chromosome replication in the S phase of interphase, just like mitosis was. Each replicated chromosome has remember, the sister chromatids that are joined at the center. Prophase 1 is super important because this is where things start differing from mitosis. Homologous chromosomes pair up, forming a structure called a tetrad, which contains 4 chromatids. You can see it right here. That's a tetrad. The homologous chromosomes line up next to one another. When the homologous chromosomes are paired up like this, the inner arms of the chromosomes, as you see here, switch. It's a process called crossing over. That exchange of information creates even more variety above and beyond the variety produced by sexual reproduction. This produces new combinations of alleles in the cell. Spindles form attached to each tetrad. In metaphase, the homologous chromosomes line up across the center of the cell. During anaphase 1, the spindle fibers pull the homologous chromosome pair apart towards opposite ends of the cell. So you see this separation happening between the homologous chromosomes. These go this way, these go this way. As anaphase 1 completes, the separated homologous chromosomes are at opposite ends of the cell. During telophase 1, there's a nuclear membrane that forms around each cluster of chromosomes, and then we have cytokinesis that forms two new cells, division of the cytoplasm. So, to sum up, meiosis 1 ends up with two cells. These are called daughter cells. Same after mitosis. Each pair of homologous chromosomes was separated, so neither daughter cell has the two complete sets that it would have in a diploid cell. The two cells produced by meiosis 1 have sets of chromosomes and alleles that are different from each other and from the diploid cell that entered meiosis 1. The two cells that are produced enter a second meiotic division. This is also really important. Neither cell goes through a round of chromosome replication before entering meiosis 2. This is how we end up with half the normal number of chromosomes, half the amount of DNA, as in regular body cells. This is going to sound pretty similar. Prophase 2, chromosomes have two chromatids, they become visible. They do not pair to form tetrads. In metaphase, the chromosomes line up along the center of the cell. This is metaphase 2. In anaphase 2, the sister chromatids separate. So you can see this looks very similar to the mitosis steps. And then telophase 2 and cytokinesis ends up with what you see here. Each of the four daughter cells in this example, and this is a simple example, receives two chromatids. And you can see, if you look at the colors of these chromatids, you have varied gametes. None of them is the same. Haploid number of chromosomes, 2 chromosomes each. And again, they're different from one another. Haploid cells are called gametes. In males, it's sperm. In females, eggs. Here's an interesting fact. 4 viable sperm are produced from meiosis in males. But in females, only one of the cells produced is involved in reproduction. Fertilization generates new combinations of alleles in a zygote because you have genetic material from 2 different parents, a male parent and a female parent. And then that zygote undergoes cell division by mitosis and eventually forms a new organism. What are the differences between, and the similarities between meiosis and mitosis? Mitosis is asexual. Meiosis is an early step in sexual reproduction. There are 3 other ways that these processes differ. It's a great table here. So in mitosis, each daughter cell receives one complete set of chromosomes. In meiosis, we end up with half of the normal number of chromosomes. The crossing over process, the sorting and recombination of genes in meiosis, result in variety. In mitosis, the daughter cells are identical. Mitosis doesn't change the chromosome number. Meiosis reduces it by half. A diploid cell that enters mitosis with 8 chromosomes will divide to produce 2 diploid daughter cells, each of which also has 8 chromosomes. We're going to use 8 as the example there. So this is mitosis. Meiosis, if we have a diploid cell that enters meiosis with 8 chromosomes, it'll go through 2 divisions to produce 4 haploid gamete cells, each with only 4 chromosomes. Mitosis is a single cell division, resulting in the production of 2 genetically identical diploid daughter cells. So single cell division, 2 identical diploid daughter cells. Meiosis has 2 rounds of cell division, produces a total of 4 different haploid daughter cells. So these are the main characteristics, the ones I underlined in red, the main characteristic differences that you need to know between mitosis and meiosis. We talked about the principle of independent assortment, that genetic traits assort independently of one another. And again, like I've said, there are exceptions. Thomas Hunt Morgan, we talked about the other day, studied fruit flies, and he came up with the principle called gene linkage. Some traits appeared to be linked together that violated independent assortment. He used a particular fly with reddish-orange eyes and miniature wings in a series of test crosses, so he mated them together. His results showed that the genes for those 2 traits were almost always inherited together. It was a rare event that you saw a fly with reddish-orange eyes and not miniature wings. They usually had both traits or neither. They saw this with so many genes being inherited together that they could group all the fly's genes into what we call 4 linkage groups. The linkage groups assorted independently, but all of the genes in one group were inherited together. Interestingly enough, Drosophila has 4 pairs of chromosomes, which were the 4 linkage groups. So, 2 conclusions from this. Each chromosome is actually a group of linked genes. And second, it's the chromosomes that assort independently, not individual genes. Alleles of different genes tend to be inherited together when those genes are located on the same chromosome. Alfred Sturtevant wondered if crossing over between genes during meiosis, when those arms of the homologous chromosomes exchange genetic information, might be a clue to the gene's locations. he said that the farther apart two genes were on a chromosome, it's more likely that that crossing over event would occur between them. If two genes are closer together, then the crossovers should be rare. If they're farther apart, crossovers should be more common. So now within the linkage groups, we're determining how close together two genes are located. So he used the frequency of crossing over between genes to determine their distances from each other. He presented a gene map showing the locations of each known gene on one of the Drosophila chromosomes. That's what you see over here, a gene map of Drosophila chromosome number 2. His method is continuing to be used to construct gene maps today.